Okay. Now, okay, here the example begins from here. How are you gonna talk about your data? So first, you have to talk about your question, your model. So the question I'm gonna ask in the following slide is to ask, does transcription and splicing are coupled in this? So I even make a slide, a single slide, to tell the audience what is the question. So everybody will make sure to follow. Okay? Now that's the question. So what is the second part? Experimental strategy. In order to address this question, we need to have a system that have transcription and splicing happening at the same time. Okay? So here is the reporter system. We put it into the cell. The science is not important. The concept is important. We have a system that we can turn on the transcription using a galactose-mediated stimulated promoter, GAL1. When this is turned on, then this gene will make a RNA that contains intron that requires splicing. So here you have a system that has transcription and splicing at the same time. So when you grow the cell in raffinose, there's no turn on of the gene. But when you switch the culture to galactose, then that turn on the transcription. RNA polymerase turn on the transcription, and then RNA gets synthesized. So we were asking that if transcription and splicing happen at the same time, splicing factor would have bound to the RNA already, happening at the same time. Okay? So then we would anticipate to see that when we do the chip assay, we cross-link the protein, this splicing factor will get cross-linked to very close to the intron. So I spent time explaining how we did the experiment. I even draw it out. My postdoc drew it out very nicely, this representative slide. And so then we go through the chip assay, we precipitate down the splicing factor, looking at what piece of DNA or RNA that comes to touch it with, and using that. So that's an experimental strategy. Many speakers did not do that. They just jump right into their experiment. And many of the time, they don't even talk about how they did the experiment. They just show you the result. And then with this graph, that graph, and you just get lost. You should spend a little bit of time talking about your experimental strategy. Even if you don't want to do this, at least you should put out a flow chart right next to your data. So you have, we started with this, that, and that simple flow chart. That will do it. And then the next, anticipate the result. Anticipate the result. So anticipated result is that when we anticipate that if transcription is not going on because the cell was growing in raffinose, there will be no RNA transcribed, there will be no splicing factor get recruited. That's anticipated. Now, if we turn on transcription by putting the culture into galactose, we expect the splicing factor will get recruited into a peak like that. This is what you're going to see in the next subsequent slides that you're going to see. You prime the audience. They know what are these graphs, what are these lines is all about. Okay, so you prime them. So that's the anticipated result. Then you can go on to describe your the data analysis. Okay, then I'll come back to that a little bit. Now, rule number 3, as I said, discuss only relevant data. Don't talk about everything under the sun. This is a common problems for graduate students. Graduate students love to talk about everything. Okay, more definitely is not better, not necessarily better. You should have to practice very well thought out minimalism. Because your time is only 40 minutes, people have to understand within 40 minutes what you're going to talk about. Don't cram a lot of stuff into your slide. Okay, this is really a common problem here in Taiwan. People they just want to, you see, similarly, the graduate students say that I have done so much work, I want to show them. But yes, you show them, but nobody understands what's the purpose of that. No good. Okay? So you have to learn how to reduce your complexity creatively. Okay? And here is something you should keep to your mind. In one slide, show only one set of data. No more than one set of data. One set of data. Okay, I'm going to show you in the next Because if you show too much, imagine that you're going to have 5 jail. 1, 2, 3, 4, 5 here on this slide. What's the audience response? Man, I couldn't understand this. This must be so complicated. That's a typical response, right? And then further, if you show too many pieces of data, what happened here is that this data will be so small. People sitting on the back row, they cannot see it. They give up. You lost a fraction of the audience already. What you should be doing is that even if you want to talk about this part, you should blow it up to occupy the entire screen. Entire screen, so that everybody can understand. Now, here is the one example. In the previous example, I showed you the experimental design, right? Then I'm going to show the data. I summarized it. I said, we change the transcription by this, by that, by yellow one, and then indeed we see the recruitment of splicing factor go down. Instead of showing them 1p, 2p, 3p at a time, I summarized them so that people can understand. And one set of data, one slide. Now, here is the common problem that they are taking out from graduate student slide. They want to do a journal club, right? Students do a journal club, and then they are too lazy. They go to figure 1, and then download figure 1 and just put it into one slide. So there's a lot of information here. So what do you have to do? Break it up is easier to understand, right? So break it up. So you're going to see a series of slides. I break it up. First, I'm going to show this one. Second, I'm going to show this one. So try to kind of have an idea where I'm going. And third one, I'm going to show this slide. Break it up. One slide, one data. One slide, one data. Okay, so here is a slide that already blow up sufficiently enough. But what I really want the audience to see is this part. So what I did, one trick. Just blow it up. Really make the things you want the audience see big, loud, and clear. Why bother to hide it in such a small thing where you have large amount of space right there? Here I can tell you a little bit my personal joke. I think you will laugh at it. When I was a graduate student long time ago, okay, so I learned how to run agarose gel. Many of you have run agarose gel electrophoresis. At that time, we don't have digital camera. We have Polaroid. Young people probably have no idea what is Polaroid. Polaroid is something called instant photograph. You take a shot, and then there was a put. There's one little thing come out, and you pour it out, and then you hang it, count 30. Then you peel it out. That's one photograph, right? That costs money. Now you can take 1,000 digital shot. Who cares, right? You can wipe them all out, and it costs more money. But every piece is money. So the senior graduate student taught me how to use that in the dark room. Then we have a big Polaroid machine right there, and then he took the gel, he put it onto the light box, turned on the UV light, and turned off the light, right? And he said, okay, what you have to do is to do this, do that, and then he poured it out, and then he left. I was left in the dark. What should I do? Should I do this first? Here first? I couldn't remember. So then the next time I run a gel, so I went to the dark room. Okay, so I tried to recall how he taught me. What should I do? So because that Polaroid costs money, I cannot keep taking the photograph. So I was nervous. So finally, I thought, I think this is what he taught me. So I tried to do that. Okay, I did take a shot. But the problem is that, imagine this is Polaroid film, and my gel is only this big. I didn't know how to blow it out. So I took that Polaroid film and back to the lab and standing by my bench and stared at it, feel unhappy about it. And all of a sudden, my advisor was standing behind me. And all of a sudden, his hand was over my shoulder. He took my Polaroid film away. And then I turned around and he said, "What the fuck is this?" And then there's a garbage can. He just threw it into garbage can. So From that instant, I realized it forever. If I want to show the slide, I want to show the data. I want to make it loud and clear, as big as possible. Fill it up. Okay, so that's one panel. Now look at the second panel. Now the second panel was cut it out from one of them. So you see what I have done is to do something. I further annotated. I annotate here, 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 here. And put it out some here, and then all these things. Because I want to make it easy for the audience to see. And I want to make it easy for myself to explain. So I further modify. Now, the third one here is seemingly very simple. But then I will be talking about this result, but mainly I'm going to focus on this area. These are control experiments. So what do I do? So I just highlight it. Okay? This is especially true. You have many, many lanes. You need to highlight the lane you want to talk about. Don't just show the jail. Okay? Now, here's another example right here. So I took it out from cell. There's multiple panels. And then some student will be presenting a slide like this. And then begin to talk about this. So, no, that's not very good. And let me just show you one example how I do it. So what I do is I take out this one. Okay? And I blow it out. But this is a complicated slide. There's a lot of lanes, a lot of bars right there. So what you have to do is that you have to use animation to guide you through it. Where you're going to begin to talk. Because if you don't think about it, when you stare at this slide, you don't know where you're going to begin to talk. So you should plan. So I would say, okay, well, we're going to talk about a protein that typically is distributed to different areas, but cytoplasm, but these are represented by 3 different colors. In the wild type, you see the distribution is blue is the highest one, green is the middle one, purple is the lowest one. Now, we would anticipate that if we perturb a particular pathway, what will happen is that this pattern will get shifted. Okay? So there is in this cell, you see here, when we delete a particular gene, the pattern gets shifted. Green becomes the largest one. You see? That helps yourself. And then further, then what you should be doing is that instead of talking about 1, 2, 3, 4, 5, because the rest of them are pretty much the same. So you just spend your time talking about this one, talking about this guy, and then the rest of them are pretty much the same. Case closed. Done. So the point here is that when you design a slide, you must think through it. Where you're going to begin, and where you're going to move to the next, and then to the end. Okay. Now, so the other way that can help audience to understand you is to use colors. Use colors to reduce complexity, creativity. Okay, to reduce complexity by color and design. Now here is a slide I use in the international meeting where I have only 10 minutes to talk about my slide. So in this particular cases, I will be talking about 2 types of cell. There's a green type of cell and there's a red type of cell. In the green type of cell, this particular plasmid is still available. In the red type of cell, the plasmid can be lost by compensated by a chromosomal mutation. So I'm going to demonstrate, okay, in my talk, that we indeed have these 2 types of cells. This is a bypass mutation, bypass essential gene. So we did a southern. But then the southern has multiple lanes, right? It's very hard to explain. Most of the people just put out the southern and orally explain it. And then everybody gets lost. So what I'm going to say is that I design it in such a way that I said we're going to analyze 2 different types of potential, 2 different types of cell, and then this one I color coded with green. The other one I color coded with red. So in the next one here, when I show the southern, I explain things only in color. So indeed, the green cell contains the plasmid, the red cell contains no plasmid. Case closed. I recall at the end of my talk, of that particular international meeting, some of my friends came to me, American friend, French friend, and then one of them told me that Say, you did a very good job. I don't know how you did it because the data can be so complicated. How do you do that? I said, color code it, reduce complexity, get the message out, central message out, that's all. Don't talk about everything. So that's a pretty simple concept. And then here's another complicated thing. So we develop a lot of mutation, and they want to look for their phenotype. The science behind it doesn't matter. So in order to talk about so many mutations, we group them. Okay, we develop a lot of yellow mutation on a particular gene product, yellow tick. And these are highlighted here in yellow, color correlation. And then there are some other additional mutations behave differently. They are colored in green. And we anticipate that those colored in yellow will allow the cells to be growing, remaining growing, where the green one, the cell will be dead. So I designed the slide this way. It's very simple and very easy to see it as a whole ball picture. Okay? Otherwise, many people tend to do what? They list them in table. One line by the other line, or leave, not leave, and no one can get it. Okay? So that's the way. So here is another way that you can drill home message you want to tell the audience. Use a little bit of the animation, a little bit high-end animation. I said, well, during transcription and splicing, the transcription and splicing are coupled, so that when RNA polymerase II is migrating, it will continue to build some platform to recruit splicing factor, the purple ball, onto it. Okay, simple animation. But that's right home. Now, next one is a classic. So I asked my postdoc former graduate student Lu to design this. I gave her some idea, and then surprisingly, she pulled it through. She did a great job. So here, I want to explain a concept that in a cell, there are 3 different machinery. So transcription machinery, splicing machinery, export machinery. In this particular RNA helicase or protein is the key components that coordinate the 3 different machinery. So I said, well, as you can imagine in the wild type cell, everything was fine, sub to the green ball, the green one is still here, and cell will be happy, they will be running very smoothly. Now, because sub2 is so important, when you take it out, then the whole machinery can no longer be coordinated. Okay? Cell will die. However, in our experiment, we uncovered something that was totally unexpected and exciting in the following manner and demonstrating the transcription and splicing are coupled. So that is, we can alter the transcription machinery, alter it, and then making the system need not to have this green protein, and cell will be still doing fine. Okay? So, in this case, they still remain engaged. And however, the cell do not like it, they leave, but they don't leave very well, they run very slowly. Okay, so these are animation that you can use. So then rule number 4, you finish your talk now. You have to have a take-home message. Okay, how do you do the take-home message? Because a psychologist studied that our attention span is only 20 minutes. Thank you very much, you are all still focusing on me. 20 minutes. And most of the people, as soon as they walk out the auditorium, as I said, they forgot who I am anyway, and what kind of things I'm talking about anyway. So what you have to do then is to drill home at the end of your talk. Always include take-home message so that they can remember it at least for a few days, to the point that they can write down on their notes, okay? And so then in your take-home message, you should finish a broad view. Okay, broad view. So how does your work fit into a broad view? Okay, project a big picture, a bigger picture. How does your work impact on the field in a big way? That is something at the very last you always have to talk about. Okay,